Hello and welcome to this A-level biology revision video on nerves and producing an action potential in sensory neurons. The first part of the nerve cell that we need to consider when talking about action potentials and where they're generated are the end of sensory nerve cells called Pacinian corpuscles. Pacinian corpuscles are one of the four major types of mechanoreceptors. A mechanoreceptor is a receptor that is going to sense some sort of mechanical input. They're nerve endings in the skin responsible for sensitivity to vibration and pressure. They respond only to sudden disturbances and are especially sensitive to vibration. The vibrational role may be used to detect surface texture. Lamellar corpuscles are also found in the pancreas, where they detect vibration and possibly very low frequency sounds. Lamellar corpuscles act as very rapidly adapting mechanoreceptors. Groups of corpuscles respond to pressure changes, for instance, grasping or releasing an object. The Pacinian corpuscles are rapidly adapting receptors that detect gross pressure changes, meaning total pressure changes and vibrations in the skin. Any deformation in the corpuscle causes an action potential to be generated by opening pressure-sensitive ion channels in the axon membrane. This allows sodium ions to influx, creating a receptor potential. Pacinian corpuscles sense stimuli due to the deformation in their lamella. The lamella is the bulbous area at the end of the nerve ending. When lamella are deformed due to either pressure or the release of pressure, a generator potential is created as it physically deforms the plasma membrane of the receptive area of the neuron, making it leak sodium ions. If this potential reaches a certain threshold, nerve impulses or action potentials are formed. Three main areas when looking at the placental corpuscles at the end of sensory neurons are where pressure acts as a stimulus, the neuronal membrane, which has a charge of minus 70 millivolts at resting state, and the fact that stretching opens sodium channels and creates a generator potential, which furthermore leads to an action potential. In its normal state, the stretch-mediated sodium channels in the sensory neuron membrane are too narrow to allow sodium ions to pass through them. The neuron of the placental corpuscle has a resting potential in this state. When pressure is applied to the Pacinian corpuscle, it changes the shape of this sodium channel. This causes the membrane surrounding it to stretch. This in turn opens the sodium channel up. Sodium ions can now diffuse across the membrane through this sodium channel. The influx of positive sodium ions changes the potential of the membrane so it becomes depolarised. This results in a generator potential being produced. In turn, the generator potential creates an action potential or a nerve impulse that passes along the sensory neuron. An action potential will then be transmitted along neurons to the central nervous system. I hope that this video has been useful. Follow the link below for a blank diagram that you can use to annotate. Feel free to go back and pause the video at any time if you need to get any of the information down. Good luck with your exams.